Welcome back to our survey of neuroscience. I'm Dr. Michael Lipton, and we are here with part 5, which will be about movement. So we're going to move right along after having understood lots about the structure and function or physiology of the brain and nervous system, how nerve impulses are moved around, how they work in the area of sensation, and now we're going to talk about how they allow us to do more than just sit still. Okay, and the goals for this particular session are going to be understanding how muscles work and how nerves are part of that. Voluntary movement, meaning when we decide to do something or plan an action, how that planning and especially control of fine movement, all the amazing things you can do with your hand, take place. And we will also talk a bit about reflexes or involuntary movement. So for starters, I just want to talk about this simple example, which is flexing your arm at the elbow. And I want to talk a little bit about sort of the minimum requirements for this to happen, to go from a straight arm to a progressively flexed arm. What we have here are some bones. The upper arm is called the humerus, and the forearm has two bones called the radius and the ulna. We have a muscle, which actually it's two muscles at the top, and that's why it's called the biceps, because there are two parts of this muscle. They're attached separately at the shoulder, and they fuse together and attach to the forearm down below. The attachments of the muscles to the bone are these gray things, which we call tendons. So now, with our muscle, tendons, and bones, how is it that we get the arm to move, which essentially means changing the position of the bones? That happens by having this muscle contract, having it actually get shorter. When we say we contract our muscles, we're literally making them shorter. When you make that biceps muscle nice and big, it's because you're shortening the muscle, and therefore it gets thicker. Now, this fundamentally occurs due to an electrical impulse. And it's actually possible to make this happen by attaching yourself to a little electrical device called a TENS stimulator, or, and I don't recommend you try this, but if you ever had the unfortunate experience of being zapped at an electrical circuit, you know that your hand suddenly jerks. And that is because the electricity is causing your muscle to contract. Now, this was shown centuries ago by Galvani, who did an experiment with a dead frog. So these are just the frog's legs hanging limp. And when he applied some electricity, which in those days there were interesting things he had to do to make this happen, but when he applied that, he was able to make the muscles contract and the frog's legs came up. Now, this was done without any nervous system being activated. This is a dead frog, and this is direct electrical stimulation of the muscle. And with that, we're going to talk a little bit now about muscles. And the muscles we're going to predominantly talk about today, really only talk about today, are the skeletal muscles, which are called skeletal muscles because they attach to the skeleton or the bones and allow us to move. But muscles can also attach to soft tissues. If you look at my face, which is moving around, I can make my lips and my mouth change in ways that make different sounds. Some of that is moving bones, like my jawbone, but a lot of that is actually just moving the soft tissues of my face, the way you're able to smile, close, and open your eyes. That's all so-called skeletal muscles, even though it's not really moving bones. So skeletal muscles are muscles that we voluntarily move, meaning by choice, you think about it and you make it move. There are other types of muscle, something called smooth muscle, which is really sort of organ muscle. Think of your intestine, which contracts to move the food along and the waste along in your gastrointestinal system. That is involuntary muscle. We can't think about whether we want our stomach to contract or not. It just happens. And there's a special kind of muscle called cardiac muscle, which is kind of in between the two of them. You obviously can't voluntarily contract your heart. It goes along beating, hopefully, all by itself. But it has similarities to skeletal muscle. Now, all that said, we'll focus on skeletal muscle, which is composed of muscle fibers. And these when we say a muscle fiber, we literally mean a muscle cell, which is composed of proteins. And those muscle cells are all of these little long cellular items that come together in a fascicle, and then multiple fascicles are put together in a piece of muscle. If you ever look at a piece of meat and you see it's kind of, you can peel these little strings off, those are not muscle fibers, they're not cells because cells are much smaller, but those are these muscle fascicles. And the neurons actually send out branches which connect directly to the muscle at something called the neuromuscular junction or the motor unit, which we will get to. Now, all of that said, muscle contraction is fundamentally about these proteins, actin and myosin or tropomyosin, where we have a relaxed fiber. You have actin and myosin in red and blue, and you can see that they're sort of spaced out. And when they contract, they essentially slide together. That is what we mean by muscle contraction. And when all of those muscle cells do that together, the whole muscle itself contracts. Now, this muscle contraction is really going to take us back to everything we learned about the synapse, because at the end of the day, the way neurons connect to muscle fibers is a type of synapse. It's not a synapse between neurons, it's a synapse between a neuron and a muscle cell. And like most synapses, this is fundamentally a site where information is transmitted chemically with a neurotransmitter called acetylcholine. That acetylcholine, when it's released, binds to receptors on the muscle side, which would be what we would call our postsynaptic location. That opens some voltage-gated channels. This should all sound very familiar from our discussion of synapses. And with opening of those voltage-gated channels, we surpass a threshold and get an action potential, but it's a muscle action potential. It's not an action potential in the nerve. So this neuron up here will have an action potential that sends a signal down the axon, which then leads to neurotransmitter release at the axon terminal. And those axon terminal releases, that's acetylcholine, will then cause the next step, which is for the muscle itself to shoot off an action potential. And the muscle action potential is what causes that foreshortening or contraction of the muscle. So, let's go back to our question. How do you flex your arm? We know that you have to foreshorten that muscle, and we know that we do that by sending a signal that causes acetylcholine release at the neuromuscular junction or motor unit. But let's talk about the bigger picture, what the brain has to do with it. And to understand this, we need to go back to our picture of the brain. When we talked about the sensory system, we colored in this gyrus, this piece of brain that I'm pointing to, and said this was the primary somatosensory cortex, where you feel touch. It is separated by a cleft called the central sulcus from what I've illustrated in green, which is the primary motor cortex. It's called the precentral gyrus, and that primary motor cortex is where the main signal from the brain out to the periphery and ultimately to the muscle happens. There's an organization in the primary motor cortex. We have the head down here, the arm here, and the leg up here. This is the motor homunculus, where the neuron cell bodies that live in the primary motor cortex, these colored areas corresponding to this green strip, that location in primary motor cortex is dependent on which part of the body is being innervated, meaning which muscles are going to be controlled. And we see that areas with lots of muscles, like the fingers and the hand and the face, have large amounts of cortex. Ultimately, we have a neuron in primary motor cortex, which shoots an axon through the white matter, right, through the brain stem and down into the spinal cord. These are very long axons. Here we're talking about the upper extremity, so it may not be that long, but if we had something going all the way to the end of the spinal cord, these axons can actually be up to 1 meter or so in length. And in the spinal cord, they make a synapse on what we call an alpha motor neuron in the anterior horn of the spinal cord. That's why it's called an anterior horn cell. So we have brain going to spinal cord, a synapse in the spinal cord, and then the spinal motor neuron goes to the muscle or the motor unit. Importantly, this is a monosynaptic circuit. There are only 2 neurons to go all the way from your primary motor cortex all the way out to the muscles. Okay, let's now go back to flexing your arm. And not just moving your arm, but being able to really sort of control and move your arm. Let's say you want to do something like, chop your karate chop down, or pick something carefully up, or press the elevator button without breaking your finger because you push so hard. How does that happen? Well, that control comes from the fact that the situation in the arm is a bit more complicated than what I showed you before. We not only have the bicep muscle, which causes flexion, we have something back here called the triceps muscle, I'm pointing to it in red, which allows you to extend your arm. The way we refer to these muscle groups, which do opposite things, as we call them, it's true that one is called a flexor muscle, that it flexes the arm. Another is an extensor muscle that extends it. But we refer to these more generically as agonist and antagonist muscle groups. Agonist is where we are contracting the muscle to cause the movement. And the antagonist is a muscle we need to relax to permit the movement to proceed. Now, it is possible, by the way, to contract both of these at the same time, something called co-contraction, which is extremely important because co-contraction is what allows you to either do what we call an isometric movement, if you just want to hold your hand in one place without it moving, or if you want to really control that movement very well, you want to be able to sort of keep the balance between the agonist and antagonist muscles. So these are important concepts: agonists, antagonists, and co-contraction. Now we say agonist and antagonist as opposed to just saying flexor and extensor, because it really depends on what you are trying to do. If the movement you're trying to do is extension, then the extensor muscle back here becomes the agonist muscle, and the flexor muscle becomes the antagonist muscle. Another thing to keep in mind is that when this movement is happening, there is something called sensory feedback, where there are sensors, most importantly in the tendons, things called Golgi tendon organs and muscle spindles, which allow your brain to understand, and even your spinal cord to some extent, to understand how much stress is on those muscles and to regulate how much flexion and relaxation we need in order to accomplish the movement that we're trying to do. Now, if we think about really complex and precision movement, so you guys probably have to do this, sit down and take one of these tests where you fill in little bubbles on a sheet. And of course, we want to do a really good job like this person did and not color outside the lines, etc. That requires very fine motor control. It also requires you to really plan out exactly what you're going to do. And this is complicated when we think about doing something with your hand, but if you think about doing something with 2 hands, like playing the piano, and getting those 2 hands, all those fingers and all those muscles in your hands to work in concert, so to speak, is really quite complex, and it requires many different nervous system components, including higher areas of motor cortex, meaning above, so to speak, the level of the primary motor cortex. These are areas that are active even if you're just thinking about movement. If you think about writing on a piece of paper, those parts of your brain are going to be active, even though you're not actually doing any movement. We're also going to integrate sensory information with the motor information. For example, we're going to need to know exactly where in space our hand and our fingers are, how much tension is on our muscle. We're going to use a concept called inhibition, where we decrease the amount of stimulation going through those nerves to the muscles so we can control movement so it doesn't go beyond where we want it to be. And then coordination is very complicated and involves areas in the brainstem and the cerebellum that get things to work together in just the right way. Now that would seem like a lot of stuff, and it is, but there's actually a whole nother layer of complexity that we need to understand in terms of the motor system. Now I'm showing you this little drawing, it's old, on the right hand side. This is one of the first patients with Parkinson's disease who was writing out her name and the date. And you can see the way, this is not because it's old, right? The reason that these letters are so jagged and irregular is because this patient had a tremor. They couldn't control their hand, and you may have seen people with tremors before. Tremors occur fundamentally because we're not able to control spontaneous movement. And therefore, there's all kinds of signaling going to our muscles that we don't really need or want. And we get this tremor, this undesired movement. The structures deep in the brain, especially the basal ganglia, something called the putamen and the globus pallidus. We looked at these a little bit in the brain anatomy section, as well as the thalamus and the brainstem. They interact with the cerebrum, with the cerebral cortex, to be able to both allow us to do the kind of moving we want to do, right? Allow us to walk, allow us to extend our hand. Simple things like that require these deep brain circuits. And when those aren't working properly, then we get problems where people literally can't move or can't initiate movement. And there are neurological disorders where patients are sort of frozen in place. They have what we call akinesia, where they can't move because of problems with their basal ganglia function. Similarly, patients with tremors like this are because we're not able to use those basal ganglia circuits to suppress the unwanted movement. Now, this has all been about what we call voluntary movement, movement that we are aware of and that we want to happen. I want to transition and talk a little bit about involuntary movement. And I want to think about various ways that we might move without thinking, so to speak. So some of this is what we call stereotype movements. When was the last time you were walking down the street and you were literally thinking about the exact way you were moving one foot in front of the other? We don't think about that, right? It just doesn't happen. And that is because these are what we call stereotype movements. Not only walking, but something as simple as standing up without falling over has to do with quite a bit of contraction and relaxation of muscles in ways that allow these functions to happen without us having to consciously think about standing up and not falling over. This has a lot to do with brainstem and spinal cord function and sensory feedback from things like muscle spindles and Golgi tendon organs that I mentioned earlier. The classic example of reflex function, however, is really the way we think about involuntary movement. And these are responses that completely skip the brain. They're sometimes referred to as spinal reflexes because they involve nerves, but they don't require any input from the brain. That allows them to happen very quickly, so quickly that in one of the classic examples where you burn yourself, and I'll go over this in just a minute, or you hit your finger with a hammer when you're trying to bang in a nail, oftentimes, before you even feel the pain, your finger has withdrawn from that painful stimulus. That's how fast it is. It goes through your spinal cord without any conscious perception, without any brain understanding of what just happened. So in this example, someone's hand in the science lab is curious or not paying attention, and he goes too close to the Bunsen burner and burns his finger. That sends through a pain receptor a stimulus back through the peripheral nerve into the spinal cord. And this is the back of the spinal cord at the top of the page, the front and the bottom. So this is where the sensory neuron comes in. And through this little short neuron here called an interneuron, it sends a signal to a primary motor neuron, an alpha motor neuron, that goes out to the muscle in the upper arm in this case, allowing it to withdraw from that hot stimulus. And this happens totally between peripheral nerves, sensory and motor, and the spinal cord with this one little interneuron. So there are 3 neurons involved: sensory, interneuron, and motor neuron, 2 synapses to give us this very, very fast response. Now I want to show you another reflex that you've probably heard of before, which is going to add a little bit of complexity that I've actually deliberately left out, so we can just basically understand the concept of the reflex. But if we think about this reflex, which you go to the doctor and he takes that funny little rubber hammer and he bangs you right beneath your kneecap. And the purpose of doing that is actually to stretch what we call the patellar tendon, which is the tendon that connects the muscles on the front of your thigh, called the quadriceps, to the top of your kneecap. By stretching that tendon, we send a sensory signal back through this green sensory nerve into the spinal cord, and we see that it connects to a motor neuron, which then goes through out of the spinal cord, goes back to those muscles on the front of the thigh, the quadriceps muscle, and causes them to contract, which causes your knee to straighten out and your foot to flip upward. This is the classic knee-jerk reflex. And the 3 elements here are the sensory neuron, the stretch on that tendon, which we call the afferent component. Afferent means going toward the central nervous system, the spinal cord in this case. And then we have an efferent component, which is the motor component, which is again excitation going through the motor neuron to contract the quadriceps. But there's another piece which I want to focus on, which is that we can see that there is another motor neuron in blue, but this one is going to the hamstrings, the muscles on the back of the thigh that normally would pull the foot down. In order for this to happen, we have to deal with our old friend, agonist and antagonist. The agonist muscle, in the case of the knee-jerk reflex, is the quadriceps. It needs to contract to bring the foot up. But in order for that to happen, the antagonist muscle back here needs to relax to allow the knee to flip up. And that last component is that the sensory neuron, the signal coming from stretching the tendon, hits a little interneuron in purple, which is an inhibitory interneuron. It causes a decrease in output through this motor neuron going to the hamstring muscle, which causes them to relax and allow the reflex to proceed. So let's now finish things up by looking at a little clinical example of brain disease. So this is an example of a man who had a bunch of motor symptoms. He had rigidity. When he tried to move his arm, it was very rigid. He couldn't move it that much. He had a tremor. His hand was shaking. He was very slow, had trouble getting going. His gait was very shuffling. And ultimately, he was diagnosed with Parkinson's disease, which is due to a loss of a neurotransmitter called dopamine, which is essential for those basal ganglia circuits, which allow us to initiate movement. That's why he was slow, and allow us to suppress movement that shouldn't be going forward. That's why he has that tremor. And what I want to show you now is that there is a very innovative way. There are medications that can potentially be used to treat Parkinson's disease, and they're variably effective. In most patients, they eventually don't work anymore. And a new technique that has been put forth and is now pretty widely used is something called deep brain stimulation, where a hole is made in the skull and a wire, which we call an electrode, is stuck through the brain into those deep brain structures. Often it's the thalamus or something called the subthalamic nucleus. And the purpose of this is that the electrode is connected, it goes under the skin, and in the chest, it's almost like a pacemaker if you've ever seen that. There's a little box that goes underneath the skin, which has a battery and some electronics, and it sends out a very low-level electrical signal, which increases the activity of these deep brain structures and is replacing the function that's lost due to the loss of dopamine. And therefore, we're able to get improvement in function, decrease in those involuntary movements, and increase in the ability to initiate movement. And patients see significant functional improvement due to the use of these devices. So that's all for movement. And next time, we're going to talk about things at a higher level, so to speak. We're going to talk about cognition, including learning and memory, motivation, reward, attention, planning, all of the things that are really very much make us human, so to speak, and have a lot to do with things we talked about earlier in the course, such as brain network functioning. So stay tuned, and I will see you again at the next session.